hand it over to Daniel Ives, who I'm really looking forward from Shift Bioscience. I'm really looking forward to hearing your presentation, because I know that you and I meant to connect at one other point. So I'm all ears, and so is everyone else.
Thanks so much, Sonia. Just let me share the screen. Okay, hello everybody. Thanks for joining me today. So what I'm going to describe and some of the work we've been doing is a path to safe cellular rejuvenation. So this is something we're really excited about. What we're doing isn't a very visual science. So just on this first slide, I have got one of the few visuals I can show you, which is we're aligning cells by their transcriptional profile as a noise reduction step. And the interesting bit is we do that so we can extract the driver biology that underlies what's going on during a time course. So just to start at the beginning, aging is a tragic and expensive process at the moment, but the real excitement is that doesn't have to be the case anymore. There are real technologies which make this scenario something that we might be able to resign to the past. And one of the most powerful technologies is cellular reprogramming. So this is perhaps the most powerful paradigm, and just some data to show you at the top to explain this. This is a time course where you express 4 factors called Yamanaka factors. And these factors are able to take an adult cell and turn it into a stem cell. They also rejuvenate the cell in the process. So the blue line is the DNA methylation age, which is our best readout for aging at the moment. And you can see after 3 days, these human fibroblasts age slightly, but between 3 and 20 days, they jettison 60 years out of the system. So 60 years to build up that aging problem, just 17 days to get rid of it. And up until about 15 days, the fibroblasts retain a memory of their original identity. So there isn't anything else like this, and it's a cause for huge excitement. Cells can do this; they can reset their age back to something they experienced a long time ago. When you express these factors in whole organisms, you can do some really interesting things. So if you express the factors transiently, so at the weekends, 2 days on, and then 5 days off and 2 days on, you can substantially increase the lifespan. In this case, an accelerated aging mouse model. However, if you express these factors continuously and not just at the weekends, you get these multipotent tumors. So they're called teratomas. It's much like a body, but without a body plan. So you get bits of hair, teeth, all sorts of things coming out of them. So this is a very powerful rejuvenation paradigm, but it's a Goldilocks therapy. So, just enough, you get the benefits, but too much, you've got something to worry about. So, it would be fantastic if we could just learn a bit more about what's going on during cellular reprogramming. So we can enrich the useful part, which is the rejuvenation, and we can jettison out the dangerous part, which is the change in cell identity. At the moment, all we know that is that there's some Yamanaka factors at the top. There's a few proteins in the middle, but mostly black box. And at the bottom, we've got a change in cell identity, and we've got rejuvenation. It's just a huge opportunity to find out what's going on in this black box, and that's what we're setting out to do. So what we've been able to do is apply machine learning in a novel way to cell reprogramming to create a list of genes derived from this, cellular rejuvenation paradigm that at first glance appear to be safe, but now we want to get into the dish, show that they can rejuvenate safely, and we can unequivocally show that these don't, cause any change in cell identity, particularly pluripotency. So to this end, we've patented the method that we use to, reach this unique vantage point. And we really think this represents the first path to safely resetting cells and tissues to a youthful state. So there's nothing to say you can't do it with the Yamanaka factors, but we think there's so much more to discover about cellular reprogramming. Why take the risk when you can increase the safety profile and potentially the efficacy just with a little bit of extra work? So that's what we want to do. We're stationed in Cambridge, UK, at the Milner Therapeutics Institute, where we're basically here for an all hands on deck experiment, testing these genes, seeing if they're as exciting as we think they are. And just at a very high level, what we're doing, so we use gene expression. So we don't look, it's actually genes. For historical reasons, we choose to look at genes. So we take gene expression matrices. So these can actually be from populations of cells, or they can be from single cells. And then we sort of distill the pathway information. So by looking at pathways, we can get a better idea of what's really going on. If you look at the individual genes, they can change in arbitrary ways. And then we use those pathways as features for machine learning. So we train the machine learning, and we ask the question, which pathways contribute most to the time course under study? This can actually be any time course, but our focus is aging, and more recently, rejuvenation with Yamanaka factors. So what we get is a list of pathways ranked by their contribution to the time course, and then we maintain a link between the pathways and the genes. So we subsequently get a list of the gene contributions. So which gene is most responsible for the changes in this time course, and then other genes follow. And this is what happens when we apply this machine learning to an aging time course. So in this case, it's the a mouse heart endothelial time course. So this is taken from the Tabula muris sanis that was created by Tony Whiskeret and his lab. So this machine learning basically creates something called a single cell aging clock. So it's just the term for the application of machine learning in this context. So we can actually with an accuracy of 0.92 Pearson's correlation coefficient, call the ages of individual cells. So you might notice individual dots. They are individual cells. So you can see all of the cells age, and the accuracy is very good. So it's above 0.9, and the MAE, so the mean average error, is less than 10% of the time course. But because we use genes, it actually gives us a huge advantage in terms of interpretability, like what's actually going on. So when we look at the gene contribution to this clock, so basically, it's the gene contribution to the time course. Is it responsible for what's going on in the time course? We see lots of positive controls. So what I mean by that, it's biology that's been previously linked to aging, or shown to drive aging phenotypes. So we see a senescent gene, we see developmental genes, we see a DNA damage response gene. We see metabolic genes, in particular mitochondrial genes and ribosomal genes. There's even a gene that's on its own is sufficient to drive progeria in humans. And this gives us huge confidence because it sort of tells us this is potentially a window into the aging biology, because we see these positive controls inside this clock. And this gives us a strong confidence that this is, this machine learning and this clock that we create is a causal clock. So it's a clock that, when you spin the sort of hands back, it actually spins back the process. And just sort of zoom in a bit, here's the genes again. Not only do we enrich causal biology, but a huge advantage of genes is that we can test their contribution to the time course rapidly. So you can, you can knock out a gene, you can overexpress a gene, and with mature technologies, it's not something that's experimental in its own right. And then once you validate a gene, you can actually drug that gene relatively easily. So you either directly target it with an mRNA, or you target the gene product with a small molecule. So genes have these advantages as far as interventions are concerned. Now, if we apply this methodology to a rejuvenation time course, and I showed you that earlier, expressing the Yamanaka factors, you can jettison 60 years out of the system, at least according to the methylation clocks, in a relatively rapid amount of time, 17 days. So we create this accurate rejuvenation clock, so 0.97 Pearson's correlation coefficient, and a mean average error of 5.7% of the time course. And then we get the gene contributions on this side. So we, reassuringly, we get genes that interact with the Yamanaka factors, which is a nice positive control. But we see, some interesting genes that you also see on the aging side. So immune genes, we see metabolic genes, including mitochondrial genes, ribosomal genes. And we see, new genes, things that haven't been linked before to aging or rejuvenation. So they represent novel targets. So, we have this list and we're hugely excited about it because many of the genes on this list don't have a link to pluripotency. They don't have a link to cell identity. In fact, we see them on the aging side and they look safe. This is the excitement. They look safe and they've been generated by the machine learning that's told us this is what's responsible for rejuvenation. So this is where we are now. We have our list and we want to take as much of that list from the clock as possible and throw it into an aged cell system, we're very fortunate to have access to a system that's able to rejuvenate from advanced stage to zero with existing paradigms in less than a week. So what this means is we can throw our candidate safe rejuvenation genes at the beginning of the week, and at the end, well, even before the end, we have samples, and then we can do methylation clock analysis to see, was this successful or not. And what we want to do is, basically identify combinations of genes and that not only rejuvenate, but they do so safely. The excitement the first time we do this experiment is actually the collision between the machine learning model and the real world. What's going to work, what's not going to work, and then we can look at the differences between the two. And that's a great lever to improve our machine learning. So we can basically go back, change the model to try and predict a more accurate list of genes, go in, see what the overlap is. And it's a bit like an eclipse. Slowly, the machine learning model moves over the real world until there's a full overlap. And at that point, the list of genes stabilizes. And for us, the therapeutic space around those genes is incredibly valuable. That's sort of therapeutic IP that we want to claim. And then the next step, the exciting step, it's preclinical development, clinical development after that. So we're going to run these cycles every 6 weeks at the maximum. Hopefully we can bring that down. We run these cycles until we basically capture the list of safe rejuvenation genes that are available to us according to the biology. This is just a timeline, with a big red dot where we are. So in the last year, it's just really sort of picked up as far as pace is concerned. We filed a patent on the machine learning method, which just applies to time courses, and not just rejuvenation time courses or aging time courses, but it could be an age-linked disease time course, and we can extract the driver biology. We recruited the inventor of the methodology, so he's a CSO now at Shift, and all the intellectual property best with us. And the next stage are these cycles that we want to run. So we don't want to just run 1 or 2. We want to keep going until exhaustion. So we don't have to come back and do this again. We basically do this, completely, and then we can move ahead. So we're going to, the end result of this is therapeutic intellectual property around safe rejuvenation genes. We're funding right now. So if you're interested in supporting us, we're raising $5 million to do this properly. And then after we do this, it becomes a question of, how do we target these genes? Do we use an mRNA or do we use a small molecule? And what's the clinical indication that we're going to select? And just to show you the pace we're moving at, so in July, we filed the method patent. In August, we dug deeper into the publicly available data sets and actually found sort of a digital version of the experiment that we originally used, which is a microarray, and that's changed the gene list. So we've got a much more robust set of genes. This special cell line that rejuvenates very quickly, nobody could get any genes into it. And we were able to crack this problem just in September. And just this month, we've been able to identify, using the same machine learning algorithm, a cell surface marker of a rejuvenation. And this just gives us a huge amount of extra efficiency, because we can select the cells that have undergone the phenomenon and characterize them much faster. Otherwise, we have to do expensive omics across a whole population where not necessarily all of the cells are doing what we want them to do. So they're not rejuvenating. And as far as deploying these technologies, there's sort of 2 sides to this. There's there's the health impact, and then there's the market size. They're broadly correlated, but they're not exactly the same. So osteoarthritis and cartilage seems like a good place to start. You can then go into muscle. Blood seems like a good thing to rejuvenate, because you could do that ex vivo, sort of rejuvenate the stem cells and go in. I have to credit Sebastian Brunemeyer with that idea. I think it's very smart. And obviously whole body rejuvenation is the ultimate. And there was an analysis by Andrew Scott and David Sinclair that put, I think, $38 trillion value on a healthy year. So we can expect at least $1 trillion market if we can do this properly. My background, I used to be a mitochondrial biologist. By trying to find out the significance of mitochondria to the whole picture of aging through methylation clocks, I got really interested in clocks. And then we wanted to develop these, single cell clocks. That turns out that they might be more than that. They might be causal clocks. Brendan was the inventor, and I sort of helped guide him to create these clocks originally. And he's the CSO at Shift. Romania was a mitochondrial biologist with me. Steve is a serial technology entrepreneur. He was in the family, helps us set this up. And Junaid's a sort of a windfall for us. He was at the Cambridge University core facility making induced pluripotent stem cells. So he's made over 120 induced pluripotent stem cell lines. He actually set up the core facility at the Sanger Institute. So it just means we can hit the ground running. We're supported by some fantastic people. Jonathan's a local angel. Carl is a US-based angel. The only person actually asking for the type of company we were building. Ken Raj is like Robin, where Steve Horvath is Steve Horvath's Batman. He's sort of behind the scenes doing the really interesting stuff. And he's been helping us sort of get to the front lines, guiding us, making sure we're working on the right problems. And Wolf Reich let us embed in his lab. We're on a bunch of accelerator programs. Most recently, the NVIDIA Inception helps us with our clocks, and Foresight is sort of opened up us to the US side. And that's my presentation. So any questions, please get hold of me, daniel@shiftbioscience.com.
Sonia, can you unmute and come online and have any questions for Daniel?
Yes, sorry, I was trying to start my video and it wasn't starting. I'm back. Wow. So that's a lot. You covered a lot. You're really going big here. And you know, there's so many things I could ask. So you mentioned earlier in your presentation that the machine learning and this whole technique could be used for really anything, but you're going after aging instead. So you're going after, it's almost like you're going after the hardest thing rather than, although at the end of your presentation, you mentioned that it looks like you're going to focus on diseases like arthritis and other diseases of aging. So can you say a little bit more about what, like, who would your customers be? Like once you get through and you identify, you've identified some of these genes, what's the next step?
Sure, yeah, so our focus on rejuvenation is just because the application is so exciting, right? The application draws you in and you want to solve these problems because the upside is so high. But yeah, this technique can just, it's designed to extract driving biology from a time course. We do want to basically find rejuvenation mechanism, genes. The idea is to use that in specific clinical indications, but there is a disease-specific component to these diseases. So if you could get a time course for, say, osteoarthritis or another disease, you could actually, precision design something for that and put it on top of the rejuvenation mechanism drug. And those two together, that's something really powerful. But we need to see, right, because we know there's a spillover from rejuvenation of age to sort of helping diseases. So Tony Whiskray with Alkahest, he's been able to affect Parkinson's disease, Alzheimer's disease with just a rejuvenation mechanism drug. So we sort of need to see how much effect size we've got, and then we might be able to sweep away these diseases at the same time, but we do have the power to sort of deep dive on the disease specific part if we have to.
And so are you looking to find drugs, or are you looking to use gene therapy? Because that was a little bit unclear to me.
Yeah, so our preference is an mRNA therapeutic just because of the speed of development. So we expect there's going to be 3 to 4 safe rejuvenation genes, because the Yamanaka factors are a great example. Yeah, there are 4 factors. You can't create stem cells with a single factor. So let's assume it's going to be 3 or 4 safe rejuvenation factors. mRNAs, you can just tweak the sequence, right, and then target the different genes and bolt on stabilization technologies and things like that. If you try to do 4 small molecule programs at once, the complexity goes up. So mRNA is our preference, but it's a rapidly moving area. So we expect it to do quite a lot on its own.
And so your trajectory then is to, find some of these targets and then start in rodents? Is that the plan?
Yeah, so find the genes, sort of design, hopefully mRNAs targeting those genes, and then, the exciting bit, and it should be more exciting because we've asked the biology, where should we start? And it's told us this is the best place to build. And then we go, into the preclinical mouse model, whatever disease, it is, we will select that model and then move forwards. And it becomes an exciting process, because the effect size should be enormous because this is the biology, right? We've asked that question. Yeah.
And let me just ask, I know we don't have very much time for questions, but let's just go back to the safe thing again. I like the fact that you're trying to make this safer. can you just explain again why your method is safer than, like, in just a couple of sentences?
So we have the potential to be safer. So we have a list of genes right now that aren't linked to pluripotency. I mean, they're far safer than pluripotency factors, which by definition, change cell identity. So the potential's there, but we need to prove this out in the dish, which is this next step, right? This is why I want to talk to everybody, like, help us find and validate these safe rejuvenation genes, because it looks like they're staring at us. We just need to prove this, like, unequivocally in the wet lab, and these experiments just cost a lot more than bioinformatic experiments. So we need to just, it's 2 years and it's about $5 million, which is, it's a meaningful amount of money, but it's not it's not ridiculous. We just want to get this right, and that's what we need.
Right. Sorry, I lied. I have one more question. What about, this seems like a perfect thing to get a grant for. Are you applying for grants for your work as well, even though you're a corporation?
I think my short answer is grants are great, but you can't rely on them, because there is a lottery dimension, and you have to trust that the reviewees are forward thinking. They believe in what you're doing. I think we have to assume the grant is going to come in time. We need to move fast. Like these things are on the list. We just need to get going now. So it's, in my experience, it's been easier to just talk to people, especially forward thinking people, and just bring them on board and start moving fast. That's been my experience.
Great. Well, I hope you're successful. I love the idea.
Okay, thanks so much, Sonia.
Thanks.